welcome to the ACSS Journal webinar series. I'm your moderator, Dr. Drought, the current science communications editor for the plant genome, and an assistant professor at the Université Laval in Quebec, Canada. Thank you for taking time to join us today. We are excited to continue this monthly webinar series, which features the latest research published in ASA, CSSA, and SSSA journals. Before we begin, I'd like to mention that the questions can be entered in the Q&A section, and we will address them at the end of the presentation. We're talking of around 3-4 questions. The webinar will be recorded, and the recording will be emailed to all the attendees after the session. Without further ado, I'd like to introduce today's speaker, Dr. Jason Gillman. Dr. Gillman is a USDA research geneticist within the Plant Genetics Research Unit located on the University of Missouri campus in Columbia. And he is an adjunct faculty member with the University of Missouri and the University of Tennessee. Dr. Gillman's research focuses on identifying and characterizing genetic factors to improve soybean seed quality and tolerance to elevated temperature and drought stresses. Today, Dr. Gillman will speak about his research using the ratio of carbon-13 to carbon-12 in plant tissue as a surrogate measure of the water use efficiency. The research report will be published in the March issue of the Plant Genome. Dr. Gillman, welcome, and the floor is yours.
Thank you very much, David. Okay, well, first off, I'd like to thank you guys for inviting me to give a talk and everyone who showed up. So I'm really delighted to have the chance to talk to you about a recent paper we had published, or we're going to have published in Plant Genome. So the paper is entitled Identification of Genomic Regions Associated with Plasticity of Carbon-13 Ratio in Soybean. I admit that's somewhat abstract, and for some of you, carbon-13 ratio won't really mean much. The subtext here is that my group is working to improve the ability of soybean to tolerate drought stress and improve water use efficiency. So as I've been talking, you've been seeing this US drought monitor tick through the year 2022. So the deeper the shade of red, this is the more intense of a drought, going from none to D0 through D4, which would be an exceptional drought. So as you probably are aware, the Western US is currently in the grips of a multi-decadal mega drought. And while soybeans, for the most part, are not grown that far west or south, many soybean growing regions in the Midwest are also experiencing increased incidence of drought stress as a result of climate change. So it's in that visual context that I'd like to start my talk. So for those of you who don't work with soybean on a daily basis, this is what a typical soybean field looks like in the Midwest. So this is near Columbia, Missouri. It's at about vegetative stage V5. All land plants have waxy cuticles that limit water loss through evapotranspiration, which was really a key adaptation for the emergence of land plants. But there's still a need to exchange gases with the environment. So on the right, you have what's termed a stomata. There are about 100 to 200 of these on each leaf on a soybean plant. So carbon dioxide flows into the leaf when stomates are open, but as a result, water also escapes. And so I'm going to introduce a concept called water use efficiency, which I will abbreviate WUE. It's simply a measure of how much material, for example, photosynthate, biomass, or seed is created as a result of transpiration, or for per unit of water, apologies. So this is really a flexible metric, so it can account for either a snapshot, a time point, or it can theoretically encompass the entire life cycle of a plant or a field. It's also extremely hard to measure at scale. So many of the early pioneers in plant physiology used pot experiments where they could accurately account for water inputs and then losses through transpiration. But those kinds of experiments are not scalable. It's just not possible to do that in a field environment for tens of thousands of plants or across locations. So fortunately, there are certain signals that we can detect in plant tissue that we can exploit to gain insights into WUE. So on the left, we have a very simplified Calvin cycle, really focused on rubisco. So rubisco is an enzyme that catalyzes the formation of 3-phosphoglycerate from ribulose 1,5-bisphosphate and incorporates carbon dioxide. So it's a critical enzyme that underlies virtually all life on Earth. And as you're probably aware, all matter exists in isotopic states owing to differing numbers of neutrons in the atomic nucleus. So carbon, for example, has stable forms of both C13 and C12, well, relatively stable. And the ratio of these has been exploited for carbon dating, which has been throughout the popular science community for a very long time. But we're not really interested in those kinds of timescales. What we're interested in is that there's a quirk in how Rubisco handles different isotopic forms. So over on the right here, we have basically a simplified transition state to products, a reaction progress. So there's a difference in the activation energy for the carbon-12 versus carbon-13 versions of the incorporation of CO2, such that there's a greater activation energy for carbon-13 CO2. And as a result, when stomates are open, plants will preferentially incorporate more C12 compared to the heavier C13. When stomates close, then they're forced to use what carbon dioxide is present. And so it's correlated with water use efficiency. So there is a direct relationship between water use efficiency and this ratio of carbon-13 to carbon-12. And this is true for many, many plants. So how is this data expressed? Well, there was a pioneer in the field, Farquhar et al., Farquhar, and he did a number of studies back in the 80s. And he focused on this term called carbon isotope discrimination, which is really an expression of the difference between the ratio in air versus the ratio in plants. So this has been proven many times in many different crop species to be inversely correlated with water use efficiency. The more discrimination, the lower the water use efficiency, the higher the discrimination, or excuse me, the lower the discrimination, the higher the water use efficiency. So many people, or much of the literature focuses on this mathematical expression. It's kind of, once something gets fixed in the literature, it doesn't change very much. But I find this is actually kind of confusing. And so what I usually focus on is a way of expressing this that's more relevant, because most people are not measuring air levels of C13 to C12. They're actually measuring plant tissue compared to some sort of a standard. So we get a ratio of the two, and this is directly proportionate to water use efficiency. So, okay, why do we care about this? Drought's just an infrequent problem, right? 2022 was just an outlier. Well, actually, no. Drought is and has been the most significant factor that reduces soybeans' genetic yield potential. So on the left, we have going from 1985 through 2020, this is the average yields across the US, with yield being on the vertical axis. So everywhere where I have it illustrated in red, that is a year where drought stress was significant in more than 20% of the field. And when we look at it in a slightly different presentation, so here we've got D0 through D4 drought, you can see it's not just an infrequent occurrence. This actually occurs every few years, and in some cases it's quite significant, and much of the soybean production area is impacted by drought. And when you actually look at the money, so these are all of the indemnity payment shares, that is crop insurance, losses from 1989 to 2014. Drought is the largest proportion of this, with excess precipitation, i.e. flooding or being unable to plant, being the next largest. Okay, all right, so drought's a problem, but we can irrigate, right? That's an easy solution. Well, no, actually that's not true at all. So in terms of soybean production, this is in millions of acres on the vertical axis, and from 1900 to 2020 on the horizontal axis. So in blue, we've got these little dots that represent how soybean acreage has increased. It was introduced actually in the 18th century, but it wasn't really grown as a row crop until the 1920s. It quickly gained a major foothold over a very short term, really. But what's interesting is that it was initially not irrigated, and even when it was, it's less than 10%. And much of this is limited to Nebraska and Mississippi. So this is not a situation that's going to change. We have depleting aquifers. We've got very high costs for diesel to pump water, and there's overall sustainability concerns. So really simply irrigating more is not an option. And in addition, there's been a number of projected climate change scenarios, simulation studies, that show, so for example, this paper here, I'm just pulling out one of many papers. This is from 2021 in Scientific Reports, where they attempt to parse out in their models various different aspects, normal growing degree days, overheating, too little or too much precipitation. The long story short is that in the aggregated impact, everywhere where you see red is a negative impact, and everywhere where you see green is a positive impact, which is really only up here at the very northernmost. I apologize for any colorblind individuals in the audience. This is not my work. I certainly would never put together a red-green chart. So long story short, there's going to be somewhere between a -25 and -75, depending on the specific region, if we don't do anything to mitigate these impacts. So this is a significant problem that farmers are facing. And I should mention, this really isn't an impact of temperature per se or reduced precipitation. We're going to increase temperatures, and there's going to be a bigger difference in the vapor pressure within the leaf versus vapor pressure outside the leaf. So this is what we term vapor pressure deficit. So now I've outlined the water problems soybean farmers are facing. Let's turn to our study as the topic of the webinar. So this is our paper going to be published in March. So this was the result of a large United Soybean Board grant-funded project, which until recently was led by this gentleman, Dr. Larry Purcell. He is a plant physiologist at the University of Arkansas. I was a statistical and genetics lead on this project since we started in 2018. I recently took over as the lead PI on the project. It's largely the work of two postdocs, plant physiologists, breeders, and geneticists. These are the ones that have been historical. There are some changes in the lineup, and we're adding additional people as we go forward. So we started in 2018 and 2019, and we grew at 4 locations. This was a field experiment in Colombia, 2 locations in Arkansas, and 1 location out in Maricopa. We did a split plot, irrigated, drought, or in Maricopa, it was irrigated and less irrigated, with 3 replications per location treatment and 205 entries, which were specifically chosen to maximize genetic diversity for drought stress tolerance. There were 4 main traits and then a couple of ancillary traits that we worked on. Today, just because of time, I'm only going to talk about C13 ratio. We also leveraged data from a previous study, which was carried out in 2009 and 2010. It was only 2 locations over 2 years, so we had a lot less statistical power, and it was also not a split plot design. 373 entries. These were not specifically chosen to maximize genetic diversity. It was more or less, we pulled samples that we thought would do well. Okay, so for the statistics and the genomic analysis, which is really what gets my blood boiling, because I love statistics and I love genetics. So we use SAXS-PROC-REG to regress mean C13 ratio of individual genotypes. And we did this against an environmental index, which was calculated for each specific environment minus the grand mean across all environments. This is something that Ebert and Russell came up with, and it works quite well. So we did a bunch of data control and a whole bunch of other things, but bottom line is there were 3 traits we got from this. One is a C13 intercept. So this is genotype dependent, and it's kind of encompassing all of our locations. We also got a measurement of phenotypic plasticity, which we call C13 slope. And then we took root mean square error as a measure of stability. So C13 ratio varied considerably on a location basis. So on the left, we've got panel 1. On the right, we have panel 2. Blue indicates the irrigated treatment, and this orange indicates the drought. This brown color, it was not a split plot design, so this was just irrigated. So to remind you, so a less negative value indicates a higher water use efficiency, and a more negative number indicates a lower water use efficiency. So what we saw was that when we imposed drought, we saw an increase in water use efficiency, and this was true across all the locations. We also had some additional locations where we didn't have sufficient drought stress, so we just had an irrigated control, but it doesn't make a pretty figure. Okay, so now I want to introduce the concept of phenotypic plasticity. So this is what it looks like when we plot out our values. So each of these represents 3 different genotypes, and we interpreted these patterns in these ways. So you can have a situation where you have no plasticity and no G by E. they're basically the same. There's no change across locations. Spoiler alert, that didn't happen. We can have plasticity, but we can have no G by E. Spoiler alert, that didn't happen either. Or you can have plasticity, it can be in terms of scale or crossover events. What we actually saw was just plasticity and G by E with scale basis. There were really no crossover events. And these three genotypes, the blue would indicate one that is highly plastic. The red is one of the ones that's the least plastic across all of these. And we put in black because it's intermediate, and it also shows off very well what this lack of stability indicates. So it's kind of all over the place in contrast with the others. I should mention we saw a normal distribution for panel 1 on the top and panel 2 on the bottom for slope, for intercept, and for RMSE, or stability. So all the details are published in the paper. I'm not going to belabor that. What we then did was we plotted slope and intercept, and we actually plotted slope, intercept, and RMSE, and the RMSE didn't really help much, so we ended up focusing on this. So slope is on the vertical axis, intercept is on the horizontal axis, and we plotted these out, and anything that's above this line here at 1 would indicate high plasticity, low plasticity would be below it. Anything to the right would indicate high water use efficiency, and over here would be low water use efficiency. So the most interesting genotypes were up here, which we used k-means clustering to identify all of the different patterns in here. These ones are both highly plastic and also highly water use efficient, and those are a very valuable group of genotypes that breeders or geneticists can exploit. In contrast, down here, these are kind of our losers, so to speak. They're not water use efficient, and they're also not plastic, so not very interesting. Same thing, procedure was carried out for the data from panel 2 as well. So in total, we identified some really interesting genotypes which could be leveraged in the future. So we then performed genome-wide association mapping studies. We used C13 intercept, C13 slope, and C13 stability, or RMSE. So there's literally dozens of programs out there which differ largely in how they handle population structure and kinship. I'm really not going to go into detail on this due to time limitations, except to kind of plug another paper that I was involved with, where we did a simulation study where we tested 8 of the most common methods that had been applied at the time for GWAS. And we identified one specific program, Farm CPU, which has been updated and is now called Blink, which had the best false positive and the best false negative rate. And this was true for 3 different plant species with 2 traits per species each. So we think that this is going to be a good program across the board for pretty much all plants. So now I'm showing you just kind of a simplified Manhattan plot with chromosome going from the start of chromosome 1 through the end of chromosome 20 on the horizontal axis. And on the vertical, this is a measure of how significant it is. It's a negative log 10 p-value. So anything above this little dotted line would be significant. Plasticity, going from top to bottom, plasticity, C13 ratio, and stability. Panel 1 is on the left and panel 2 is on the right. So we found a lot of hits. Interestingly, we found a lot fewer in our panel 1, which had more statistical power than panel 2. But we did find some very interesting hits, and we looked at the literature, both our previous studies and other studies, to see if there was any overlap. So we didn't really see any specific SNPs that were identified between our 2 panels, but we did find some coincident regions with previous studies, or they were within LD. SNPs in panel 1 and panel 2 were in LD. So plasticity is in blue, C13 ratio is in green, and stability is in sort of this pinkish purple color. And these are plotted out. Every place where you see a black circle and triangles, that indicates that it's coincident with previous studies, ours or others. Full details are in the paper. So we're pretty confident that this is repeatable. We're pulling up really significant marker associations. And there were a couple that we found very interesting gene candidates here. So specifically, I'm going to draw your attention to chromosome 13 and this region here on chromosome 19. So this is a bit of a wall of text, and I apologize that. We basically identified a large number of loci for panel 1 as well as for panel 2. We found 8 loci were within the same LD block as previous reports in panel 1 and 14 in panel 2. So we identified 45 candidate genes and 29 candidate genes in panel 2. And some of these were identified based on their annotations. They were suggested to have a role in drought stress. So some of these were quite interesting. So this one for plasticity on chromosome 13, it's extremely close, if not inside a gene, which has been annotated as an auxin efflux carrier protein. So auxin's a phytohormone, and polar auxin transport is involved in responses to drought in many, many plants. So overexpression in rice of the direct homologue led to improved drought tolerance and drought stress responses. So we're quite excited about that. We had a couple of other ones which were involved in ABA, either signaling or responses, and in many other organisms, not necessarily soybean, overexpression of these has resulted in drought stress tolerance. So although the results themselves are sort of, we find drought stress tolerance, we found interesting patterns of drought stress tolerance, there will still need to be breeding work in order to really capture this going forward. So with that, I'm happy to take any questions. And thank you for attending, and thank you for your questions.
Thank you, Jason, for this nice presentation. And now we are open for the questions, and ask the attendees to enter your question in the Q&A section, so we can go over them one by one.
Okay, I do see one. It's from Kishore Gali. In this study, vegetative tissue of soybean is used for carbon isotope analysis. What are your thoughts about using harvested seed? Excellent question. So we actually, Larry Purcell and a number of others, I was not involved in this study, did an evaluation, several grow outs where they looked at the correlation between vegetative tissue, various different stages, whole plant, specific leaves, specific leaflets, and seed, and they actually saw very significant and strong correlations. We're doing a follow-up study right now where we're doing it with a little bit more statistical power, but I'm fairly certain that the correlations are going to be on the order of 0.6 to maybe up to 0.9. So long story short, you can use seed instead of having to do this extremely laborious field collection.
Thank you, Jason. We have one question from Ishwan Reshkan in the QA session. Excellent work and presentation, Jason. I missed the difference between panel 1 and 2. Could you please elaborate on that?
Oh, absolutely. So the difference was, so panel 1 was something that we did later. The panel 2, it's kind of counterintuitive. We did that back in 2009 and 2010. So that was shortly after the entire soybean germplasm collection had been genotyped using the 50K SNP. So we essentially went in and just pulled out maturity group 4 entries, which are appropriate for our growing regions, and tried to maximize genetic diversity, grew those out, and did those set of experiments. When we had all that data and we analyzed it, we then took that data and used it to identify 100 genotypes for panel 1, which represented pretty much all the genetic diversity from panel 2. And then we used a genomic selection approach to identify another 100, which maximized genetic diversity for all of our traits. So panel 1 is essentially taking panel 2 data and then improving it to increase the amount of genetic diversity.
Wonderful. Another question is from Mary Couturi. Why do you think PharmCP is performing better than other methods for GWAS?
Well, we did it empirically. So we did a simulation study, which was based off of 3 real-world data sets. And in this case, we basically designed it so that we knew exactly where the significant marker associations were, and ran it through several iterations, did it for 3 different species with 2 traits each, very different heritabilities. And then we literally just went through and said, okay, what's the false positive rate? What's the false negative rate? And across the board, PharmCP performed better. So that's the long and the short of it. It's an empirical determination. There may be other programs that we haven't tested, which could perform better. I realize that PharmCP has kind of got this weird, quirky, interactive way of dealing with kinship and population structure that makes some people kind of uneasy, but it works.
Thank you. We have another question from Laurie. Would or will CRISPR gene editing technology be useful in targeting potential drought tolerance enhancement genes? to facilitate more rapid incorporation of drought tolerance?
As long as you have a target, yes, absolutely. And as long as it's a major effect one. For the most part, for all of our drought traits, we're actually seeing really quantitative inheritance. So it's not like there's a silver bullet or one specific locus we can target. And the other part is that just because you do a GWAS or a QTL mapping study, it doesn't mean that you necessarily identify the gene. It's maybe a bit of a disservice. It's kind of the nature of science where we have to put our best foot forward and say, these are our candidate genes. But in a lot of GWAS studies, we're getting a big, broad window. And so we're taking our best guess based off of annotations, but we could be very wrong. And in many cases, looking back, we actually have been wrong.
All right. Another question from an anonymous attendee. Can you share any interesting phenotypes associated with the high water use efficiency and highly plastic genotypes?
I can't right now. I'm sorry. We are doing some studies right now, but I think my fellow co-authors would murder me if I talked about that. So no, I'm afraid I can't. Right now, the data is published. If you'd like to reach out, all of these genotypes, I should mention, are part of the USDA collection, and they're freely available for anyone that's interested in ordering them.
Another question from Zulian. Thank you for the great talk. The phenotypes on the top left quadrant have high plasticity, but low water use efficiency, right? Is that plasticity for water use efficiency only? If yes, what would the benefit be?
Okay, so yes, the ones that are in the upper right quadrant are going to be the ones that are the most plastic and the most water use efficient. So that is when water is plentiful, they have low water use efficiency, but when water is limited, they have high water use efficiency. So this is the ideal genotype that you want. The other ones in the other quadrants, particularly the bottom left, those ones are the worst. So they don't change. They basically waste water all the time. It may be that those are ideal if you're just managing optimal environments, but in any place where things are going to vary, i.e. the real world, they're not going to be ideal. And the other two quadrants are either water use efficient or plastic, but not the other.
Wonderful. Another question from Ishwan Rashkan. What was the average amount of variation explained by single significant market trait association in the GWAS?
Oh, I don't know if we've actually calculated average, but it was on the order of, I think the highest was about 6%. For the most part, it's a very small amount that's explained. The one that was for plasticity that was the highest, I think that explained about 13% of the variance, if memory serves.
Okay, great. Another question from Raju. Thank you. Nice talk. Was there a relationship between water use efficiency and yield?
We didn't actually measure yield in our studies. This was designed to be a very large experiment already. We were collecting massive amounts of tissue. We ground for months and did all of that kind of stuff. And to be honest, measuring yield on plant introduction lines is not the best use of your time. I do have other experiments that we're carrying out or which are planned for the future where we are going to look at exactly those questions. From the literature, it depends on the environment. So if you've got a place where you've got drought stress that's imposed, you're going to see higher yields associated with water use efficiency in drought locations, but that may not be true in well-watered locations. So it's really kind of, you don't necessarily always want to have water use efficiency. You want to have a plastic response.
Okay. We are going to finalize the seminar with the last question from Mary. With as many QTs as were found in this study, do you think these traits would be handled more efficiently in breeding using genomic set prediction? Yes,
absolutely. That was the whole point of doing this, was to develop valuable data sets that we could use for genomic selection. And now that I've taken over as lead PI, we spent several years building up populations. We're doing field experiments now and in the future. So we're hoping that we can prove that genomic selection works as well, or maybe even better, for water use efficiency and our other traits as well, as it has been shown to work for yield. So yes, I do believe that genomic selection is the only way to manage this number of loci.
Wonderful. Thank you, everyone, again for joining us for today's webinar, and to Dr. Gillman for sharing his research. It was wonderfully good and nice, great talk. As a reminder, the recording will be made available and sent to all attendees soon. Thank you again, and have a wonderful day. Goodbye.